that both Matt Farthing and myself have really been working pretty hard on together over a couple of years. And the authors I have over there are definitely not all the people that have been involved in the project. These are just the people that really helped initiate the project. And this is almost like a two-part series in this talk. The talk that I'm giving now is going to be more based on sort of the results and not our results, more about our methods in terms of how we got the data and what we want to use the data for. And then Matt's talk, which is later in Ben, is really talking about our results, the modeling, the meta-analysis, all the deep results. This talk is more an overview of the data we collected, the structure of the data, and what sort of patterns we saw before we conducted the deeper analysis. So pursuing the magic number, estimating species-specific post-release mortality in marine recreational fisheries through a systematic review. Okay, so recreational fisheries are complex, multi-species fisheries. I think we've heard that a lot throughout the week. They're hard to manage. Recreational fishes are really dispersed. They're large, they're valuable, and they're really impactful, and they can be used to uplift communities and are important to a variety of sectors in the economy. But sometimes, especially catch and release, is used by anglers as like a social responsibility that they aren't affecting fish stocks. But we do know that there are impacts of catch and release is not benign. Some fish do die, and while that can be mitigated against, it's really important to understand how many fish are essentially succumbing to mortality after the catch and release event. But post-release mortality depends on a suite of factors, from the specific biology, physiology, the genetic stock that's being harvested, the environment, fishing techniques, what type of hooks are you using, the gear types and fishing platforms that people are fishing off, as well as angler behavior and their own practices themselves. So it's a global problem. It's not just restricted to South Africa, and it's not only restricted to anglers that are fishing for pictures and releasing their fish. In a lot of angler legislation, undersized fish need to be released, not only by recreational anglers, but commercial anglers and small-scale anglers. So information derived in this project is applicable to a variety of fisheries as well. But there's limited data on recreational fisheries biomass removal, especially in marine recreational fisheries. And harvest is only one part of that removal. Obviously, post-capture mortality and post-release mortality often remains unknown, and there's very selected studies have been done, and those studies are still in quite limited numbers globally. And in certain instances, harvest through both retaining catch in recreational fisheries as well as post-capture mortality can rival commercial sectors. So it's really important to understand what is being taken out of a recreational fishery, especially in context where you have conflict. Like I know there's a lot of conflict at the moment in Western Australia from chatting to a few guys. So it's really important to accurately collect this data to estimate what recreationals are catching and what is dying so that, so that compromise can be made around the table. But a poor understanding of post-release mortality hampers effective management, such as stock assessments, conservation strategies, and the social license to practice recreational fishing. So in terms of previous catch and release effects reviews, the real seminal paper is this by Bartholomew and Bouzak. And these are really good reviews, but they often are really, they're reviewing the literature, they're seeing what studies have been done, and they're looking at factors that are increasing mortality, things like hooks, like what type of hook, handling, all those kind of things, where this project really seeks to actually understand what that value is, the magic number, how many fish are dying in a specific fishery after they're released. So we conducted a systematic review through three scientific paper repositories. These are the keywords we used, and we collected a lot of papers, because obviously there's a lot of overlap, and a lot of these terms will come up in all sorts of things from medical research, but it's really a big chunk of data, a lot of different papers that we had to really go through to try and close down what we needed to review. So we used in terms of our methods, so after we did that search and we got big chunk of data, a big chunk of a lot of papers. We had to go through those papers, remove duplicates, etc. And this R package, actually really good in terms of automating that process. So if you are going to conduct a systematic review or meta-analysis for your student, it's a really good package to know about and to learn to use. It's very user-friendly. So in terms of some of our results, we used the Prisma approach. So you can see, as I said, from the initial search, we got 4,800 papers. Duplicates were removed. It went down to 3,000. Then we screened records, and we excluded records that were not recreational fishing related. We came up with 596 papers. Now this includes both freshwater and marine. Then we screened these records, and we screened out everything that wasn't marine or estuarine recreational fishing. That updated the results to 275. And then we screened the results further because we're only interested in studies that reported post-capture mortality, where a lot of studies just looked at health and survival and didn't look at mortality. That number has significantly dropped, and the total database that we utilized was 106 publications. And from those 106, where they were lethal, where post-capture mortality was estimated, there were 164 unique post-mortality estimates, because certain studies will work on multiple species or conduct multiple assessments in different areas and published in one paper. So basically, from 106 papers, we got 164 unique post-release mortality estimates for 102 species across 36 families and 19 countries globally. So after we got all those papers, we screened the papers primarily for, obviously the response variable was post-capture mortality, which was captured as a percentage or a proportion of the fish that were assessed. And then we also scraped about 30 covariates that were associated with that data from these publications. And that's just a list of some of the data we scraped. Again, now moving back to results again, this is just a representation of where these studies were conducted globally. You can obviously see that, the US is definitely fairly high up there in terms of producing publications. Australia is pretty close behind, followed by sort of, if Europe was an area, it would probably be quite similar in Australia in terms of the amount of papers that produced. It's just, obviously geopolitically, you've got to report it in individual countries. Been a few studies in South Africa, and then some coming out of Brazil, and then some also in Japan. So from this data, you can see that post-capture mortality estimates have, well, at least studies have steadily increased over time. And this little anomaly over here, this uptick over here can be associated with the Boonzock paper that was published in 2005, and then it's steadily been increases. This is cumulative increases, okay? So importantly, we identified, and Matt's going to speak about the model that we used and the actual post-capture mortality estimates and and the tool that we've been working it to produce. But importantly, this is just showing the type of data that we were collecting and especially the method, the method of post-release mortality studies. So importantly, a lot of these studies were field studies. Some of them incorporated both field and lab studies. And then there were obviously certain studies that were only conducted in the lab. And in terms of scientific method, it was definitely, fish that were captured and then placed in a cage and were assessed were the highest, followed by in a holding tank. So that's outside of the water, that's in the water, in the ambient environment. And then third is acoustic telemetry is starting to be used a lot more to infer. Archival tags, direct observations, which is, for example, a study where a fish will be caught and then monitored either visually or through like an attached float. But there were also studies that utilized things such as hyperbaric chambers to understand the effects of barotrauma. And then importantly, there were in a low number, but a few studies that used a combination of a variety of results to find their estimates. And then importantly, leading on to the next slide, most of these studies were conducted from a boat. You can see very few from shore, obviously some in the lab, and then some were collected both from boat or from the shore. So leaning on that boat side of things, obviously fish that are caught from a boat, caught in deeper water, often express, they have issues with barotrauma, which is obviously of concern to managers. And given that, so when you go to Matt's talk later, he's going to talk about this barotrauma issue and how it really affects the model, and it's really important to acknowledge. But as you can see with the studies, that these are just the methods used from studies that basically that looked into barotrauma. So descending was the one barotrauma mitigation factor that was looked into the most, followed by venting, variety, and recompression. So the number of studies conducted on catch and release effects has greatly increased with time. A variety of methods have been used to derive these studies. Please ignore that 10 to 35%, because it actually goes all the way from 0 to about 60%. And boat-based field studies definitely outnumbered land-based studies. The metadata and the raw data from trials really needs to be accompanied in these studies, because a lot of the studies we reviewed didn't actually have a lot of the metadata associated with it, so it was quite hard to include it into the meta-analysis. We had to do some back calculating, etc., which really increased the time of reviewing these papers. And it's also really important to design studies that are real-world scenarios, that are really representing the angling body, using anglers, not influencing them. But at the same vein, it's important to also understand key factors that are affecting mortality, and those need to be done in the lab and need to be more controlled scenarios. So we need a suite of studies done. While it's really important to get real-world scenarios, to understand changes in methods and changes in fish catching technology, we need to also conduct more controlled studies. And then in terms of the most defensible estimates, comparisons between methods, so sort of between methods that are the same, are really good and really defensible to managers, etc. But models are better informed when species-specific post-release mortality estimates are derived from multiple methods. So while it's, I think as the community, we need to carry on doing what we're doing in terms of figuring out these post-release mortalities using a variety of methods, but at the same time, it is really good to compare apples with apples rather than apples, pears, and bananas. But with complex modeling processes, adding apples, bananas, and lemons into your model can really paint a really nice big picture. But please, for the real results in terms of the modeling, the tool, and everything is going to be reported by Matt Farthing and Ben McCullough at 2:30. So if you found this talk interesting and really want to find out the full results of the study, please go join his talk. It was just such a big project. It was too much to cram into one talk, so we sort of split it up like that. Cool.